Hello viewers, I am Professor P.K. Kathal from Department of Applied Geology, Dr. Hari Singh Gaur University, Sagar, India. I am going to deliver a series of lectures on applied micropaleontology. The topic of my today's lecture is Applications of Microfossils, Part 2, a detailed account. In Part 1, I have shown you that microfossils may be broadly divided on the basis of composition of wall. The first category is mineral wall microfossil, including Foraminifera, Ostracoda, calcareous nanoplanktons, diatoms, radiolaria, conodonta. And the organic wall or paleonomorph microfossils include dinoflagellate cyst, pollen, and spores. In this linear uh, diagram, I have tried to show the relation of wall composition with the microfossil group. Microfossil group are divided into mineral wall, organic wall. As I said, organic wall has dinoflagellate and pollen and spores. Mineral wall has calcareous, siliceous, phosphatic, and chitino-calcareous compositions. Under the calcareous variety, there are two groups, Foraminifera and calcareous nanoplankton. In siliceous variety, we have radiolaria and diatom. Under phosphatic variety, we have conodonta, and under chitino-calcareous, we have Ostracoda. Now we will see the groups, important groups of microfossils one by one. The first is Foraminifera, that is a protozoan, unicellular, calcareous or agglutinated. It is marine except a few species in freshwater, and it belongs to both planktic and benthic habitats. In the picture, I'm showing you the living planktic Foraminifera, which is calcareous, and this is the dead shell of planktonic Foraminifera, which is clearly showing that the chambers has a lot of volume, and the overall volume of the test is proportionately greater than the weight. Therefore, it should float on the surface, and its morphology is showing it is planktonic Foraminifera. On the other hand, and the picture at the bottom shows a very heavy test of benthic foraminifera, which is thick and heavily ornamented. In the second column, the top picture is showing the agglutinated foraminifera, in which the pieces of sand with other grains, black colored iron grain or some other minerals like mica flake are visible. And the second picture, which is the color SEM, is showing us that the composition of the agglutinated particle is different in agglutinated foraminifera. And the other picture is showing tubular agglutinated foraminifera, and the picture on the right hand side top is showing the assemblage of planktonic calcareous foraminifera, where you can again appreciate that the volume of the test is more. In the bottom picture, a glass jar is shown to you, where a culture of benthic foraminifera can be conducted in a very small space. Uh, Foraminifera are very useful in depicting the paleo environment of deposition. For this, there is a model called Triangular Model for Environmental Interpretation given by Morey, in which we use the ratio of three important suborders of Foraminifera on the basis of their wall composition. The first is Miliolina, which is having porcelaneous test. Second is Rotellina, having calcareous perforate test or hyaline test. Or third is Textularina, which is having agglutinated test. What we do, uh, we count the percentage of these foraminifera and we plot in this triangular diagram, and we can see that the cluster of points of different environmental condition will fall in different regions. For example, if the samples are from hypersaline lagoon, they will fall in this linear tract uh, inside this linear tract in the triangular figure. If they are from normal marine lagoons, then they will fall in this region within the triangular diagram. Similarly, if they are from the hypersaline marshes, they can fall anywhere in this white portion. If they are from normal marine marshes, they will fall within this region, inside this region, means inside this region, anywhere. And if they are from most shelf areas, they will fall within this region. And finally, if they are from the hypersaline lagoons, which is the ideal condition for various um, petroliferous sedimentary, particularly of tertiary age, where we get a lot of oil. So, in this way, if we analyze the rocks for foraminiferal content and we plot it in the triangular diagram, the cluster will show us which condition was prevailing and how the conditions changed in geological time period on the basis of the borehole log. Uh, I continue the oil exploration part. In fact, the abundance of microfossil, particularly foraminifera, is very important because we can get lot of specimens, thousands of specimens, even from small core samples, and they have a capacity to demarcate or show us and help us in demarcating oil zones to minimize the exploration cost. Being small, mostly unicellular, and sensitive to minute environmental conditions, they help finding rocks vulnerability for oil in the following ways. Low oxygen species show chance of oil formation. Agglutinated calcareous ratio, which is helpful in demarcating outer and inner bay facies in estuarine sediments for oil exploration. And elongated morphological groups show shallow, calm water condition, which can be considered good for conversion of organic matter into oil. Another important aspect is determination of paleotemperature of water on the basis of oxygen isotopes. We know that there are two isotopes of oxygen. Lightest is, there are other isotopes also, but the lightest is O16 and the heaviest is 18. So ratio between 18 and 16 is a temperature dependent ratio. The calcareous planktic foraminifera as well as benthic foraminifera, they secrete calcium carbonate and they take oxygen from the water. Under the normal circumstances, the in the non-glacial period, the evaporation takes place in the ocean surfaces, and that allows release of the lightest isotope, that is oxygen 16. And but the oxygen 16 comes back in the form of oxygen cycle, or through the oxygen cycle by rains or water from the rivers, and a delicate balance is maintained between O18 and 16, and this is depicted as zero point 0.0 under the normal condition. But under the glacial conditions, evaporation of O16 does take place, but it gets locked in the ice sheets and does not get back to the ocean. And the level of ocean, therefore, is decreasing, and there will be uh, increase in the ratio of O18, and it may become 1.5 or even more than that. This phenomena is shown in the log here, where this point is the zero. It means that it is the normal non-glacial condition, and this is the ratio of O18 is 2.0. So if the data taken from the log shows that the curve is more towards zero point, then this condition is indicating less ice and interglacial stage. But here we find in the lower part of the curve, it is more towards 2.0 in favor of isotope 18. Therefore, it is showing more glacial or more ice condition or glacial environment. So here we can make use of the foraminiferal test for calculation of paleo temperature of water. Planktonic foraminifera are very useful in biostratigraphy. It is because their distribution is so wide, and most of them are having very small ranges of their living history. Therefore, if we use them for in biostratigraphy, we can correlate it at global level. So you can say planktonic species are used in biostratigraphy and correlation at global level, especially in oil exploration and study of deep offshore environment. Similarly, appearance and disappearance of marker species from Cretaceous to recent times also help making fine biogeneration. Foraminifera in very normal way can show the environment of deposition, and it is because on the basis of population and wall composition that can show the depth and temperature. Normally calcareous forms indicate normal temperature, shallow or intermediate depth of water, and low latitudes, whereas agglutinated forms show below normal temperature, high depth, and high latitudes. So if we get more calcareous form, it is in favor of normal temperature, and if it is having very high percentage of agglutinated form, either it is because of the depth or it is because of the temperature consideration. Similarly, another important aspect is dissolution of calcium carbonate, particularly in planktonic foraminifera. If planktonic foraminifera cross these calcium carbonate compensation depth or carbonate compensation depth, the effect of dissolution can be shown. Here I can enlarge the picture and you can clearly see the effect of dissolution, and that is because it has crossed the CCD that varies from 5000 meter near the equator to as low as 450 meter near the poles. Foraminifera assemblages can show the environment and microenvironment of deposition of rocks and sediments in a basin. There are different characteristic assemblages of each environment, and I will show all the slides one by one. This slide is showing assemblage from the marshes. In this slide, the assemblage is from inner shelf area. The, this assemblage is showing that it belongs to outer shelf area. Assemblage of benthic and calcareous species in this particular slide is showing bathyal condition. Assemblage here, that is consisting of agglutinated foraminifera, is showing abyssal conditions of deposition. Calcareous assemblage from deep sea will look like this, and agglutinated assemblage from deep sea will look like this. Similarly, if foraminifera are from reef environment, there is a characteristic assemblage which is shown over here. Other important uses of foraminifera may be seen. One is morphology of benthic species. We can divide them into two groups: elongated test and rounded test. Elongated test belong to calm water condition, and if they are in good number in the assurine sediment, the chances of conversion of oil are better. But if the majority is belonging to robust and rounded type of form, it shows that environment of aeration or high oxygen environment, and therefore the chances of oil formation are meager. Hydrogen ion concentration of pH can be depicted by foraminiferal test. The normal pH in ocean water varies from 7.8 to 8.3, but with the depth, carbon dioxide concentration increases and water becomes acidic. So we will find that a small specimen of same species can survive better in acidic conditions as compared to the larger ones. Here two examples, Trochamina and Anomalina, are shown that can tolerate pH up to 7.2. The second largest group after Foraminifera is Ostracoda, which are belonging to phylum Arthropoda. and they are multicellular or animals, bivalve like lamellibranchia. Compositionally, they have chitinocalcareous walls, and they belong to marine as well as freshwater, and they can be found as planktic and benthic in both the conditions. They have got a lot of utility in oil exploration. And the special thing about Ostracoda is that certain parameters can be worked out on the basis of Ostracoda without going for their identification. For example, carapace and wall ratio can show the rate of sedimentation. If the walls are closed, maximum walls are closed, it means the sedimentation did not allow time for opening of the valves, and that shows the high rate of sedimentation good for formation of oil. Similarly, the color of ostracoda, if it is changes to dark brown or it becomes pyritized, then it shows the degree of reducing condition, and chances of transformation of organic matter into oil are better. Current index can also be studied on the basis of normal ratio of juvenile and adult carapaces, that is 7 is to 1. And if it is 7 is to 1, then aeration has not affected it. But if it is disturbed, it means more aeration and less chances of formation of transformation of organic matter into oil. Compaction ratio is important for formation of oil pool. So if they are deformed ostracoda, they show the degree of compaction, and on that basis we can decide with the migration of oil in the form of pool in the nearby areas. Calcareous nanoplankton are very important group of microfossil. There is their specialty is that they are very small, nano, and they range in size from as low as small as 0.25 to 30 millimicron. Is very small size, and we need a scanning electron microscope for their identification. They are the planktons and floaters in the upper layer of ocean. Collectively, there are three groups called nanoliths. They include Coccolithophyceae, which is the living algae, only living form, and discoester and nanoconate. The discoesters and nanoconates are the extant form. Coccolithophore is the living member that belongs to superphylum Protista, that include unicellular organisms with nucleus, phylum Chrysophyta, because they have golden brown color pigment of chloroplast, and class Coccolithophyceae, because they can form cocosphere or coccoliths, and because they can build a rock. Therefore, the name Coccolithophyceae has been was proposed. Calcareous nanoplanktons, as I said, have three groups. Coccolithophyceae range algae range from in size from 3 to 15 millimicron in diameter, and they wear shield-like buttons which are discarded upon its death or also during the lifetime. And here in the picture, you can see that this is one cocosphere here. This is another cocosphere, and there is an envelope of overlapping shield-like structures which are called as coccoliths. So here you can say that see that one coccolith has been enlarged, and if the calcareous nanoplanktons are there in carbonate rocks, the thin section will look like this. The second group is discoester. Their name is because they are star-shaped. I have included two species for your ease here, and they range in size from 5 to 25 millimicron, and they are asterisk shaped. The third group is nanoconate. Nanoconate means small conical wedge-like structure which may collectively form a flower-like pattern. They range in size from 0.20 to 30 millimicron, and they can look like a very miniature flower-like structure, and they do not have any clear lineage with any living organism, and these discoesters and nanoconates are the extinct groups. Use of coccolithophore or nanofossils are many. They are used in high resolution biostratigraphy of Mesozoic and especially Cenozoic time. And let me tell you that the International Telescope Time Scale incorporates the biozones on the basis of calcareous nanoplanktons, particularly in Cenozoic time. The study of surface water paleotemperature is possible with them. They also show they have also shown shift in Miocene water masses, which is very important because we know that in Miocene time, circulation of ocean current, ocean water has changed to a great degree, and they can also show us the glacial interglacial zones, particularly in Pleistocene time, on the basis of O18 and O16 ratio, as we have discussed in case of Foraminifera. They can also show the chemistry of surface, column, and bottom water, and also the rate of sedimentation. Organic wall microfossils include pollen and spore. They are resistant to microbial physicochemical attack after burial. Therefore, they occur in very great number in the rocks, particularly in the near shore or coastal areas. They are most abundant and widespread, most widely by wind and water. Ecology-based data gives us paleo environment of deposition. They show distance to land in marine rocks, and they can be used in biostratigraphy as markers in lake, river, and deltaic rocks, or in the study of paleolimnology and correlation of continental and nearshore marine rocks also, particularly from Devonian or younger ages. In the pictures, I have shown you one pollen which is nearly triangular in outline, and here there is a cloud of pollen released from the male part of flowering plant, that is called androecium, and they are released in millions and billions and sometimes in trillions by a single tree, and their rigid outer surface help them preserved in the rocks, and therefore their abundance is very high. And in the lower picture, the it is showing the spores, germinating units of lower plants, which are also released in a very great number, and they are also rigid and can be separated from rock and can be studied. The other group is dinoflagellate, which is unicellular cyst preserved, and or they may be organic and calcareous or siliceous, and they may be found in marine as well as freshwater. They are useful in biostratigraphy of marine Mesozoic, Cenozoic rocks with Acritarchs and pollen spores. Study of provenance and direction of transport through reworked cyst and spores is possible because they have rigid outer surface. Kinds of cyst can show near and offshore strata, and quaternary cyst groups are also there. Recognized groups are there. They show the probable climate control, correlation, and interpretation of marine strata. In the picture, this is the living cell of dinoflagellate, and this is one handmade sketch. Diatoms is another important group which are siliceous and belonging to unicellular plant origin autotrophic algae, and it is important because they occur in marine as well as freshwater. So in freshwater they are the only siliceous hardcover forming algae, and therefore they are very useful in the study of sediments, study of sediments, paleolimnology of most of the quaternaries have been done on the basis of diatoms. They have got various uses, like primary means to correlate high latitude deep sea sediments, and they first appeared in early Jurassic. So any rock is which is consisting them is not older than early Jurassic, but they became abundant in early Cretaceous only. Biozones in Cretaceous and Tertiary interstitial sequences are important. They are valuable for not only warm water but also cold water biogenation. Paleocological value in Pliocene Pleistocene is very high. Silica test can give us paleo temperature on the basis of O16 O18 ratio, and freshwater diatoms can help developing the history of lakes, acidic effects, and to the climate or anthropogenic stress. And in the picture, I have shown two types of diatom. One is centric diatom, which is circular in outline, and second is the pinnate diatom, which is having leaf-like shape. And you can say a lot of pores are there on both the surface, the surface of both the types. And here there is one rock entirely made up of diatom, that is called diatomite, layered sedimentary rock. And this diatomite is used nowadays in the industry for filtering sugar and making good quality toothpaste, and also as insulator and polishing material. And the modern species of diatoms are used in forensic investigations in a very high way. Radiolaria are another important group belonging to protozoa. They are cousins of Foraminifera, but they make siliceous test, and they belong to very deep marine water where generally we do not get any calcareous species. The reason that the calcareous forms they get they are dissolved below the CCD, so they are the only indicators of the deep marine water conditions. Their biostratigraphic value is very high, particularly in joining of Mesozoic rocks, where other calcareous forms are not found. Their architecture allows showing minute evolutionary changes, and evolution correlates well with the magnetic reversal event of the last 3 million years. So the stratigraphy developed on the basis of radiolaria go hand in hand with magnetostratigraphy. I have shown some pictures of radiolaria. Conodont is another important group of early Paleozoic and Middle Paleozoic time. They are the small chewing parts of a large set found in an eel-like animal called Odontogryphus. And in the picture, I have shown the two elements in the upper part, and the set of or apparatus of conodont of Odontogryphus is shown in the lower picture. They have got a lot of biostratigraphic use because they are exclusively marine, or rather they were exclusively marine. So only thing that we know is in the form of paleo history of their living, and they can be used in the joining of Ordovician mostly, and also sometimes in Permian and Triassic marine rocks. They in a way they support sea floor spreading, and degree of change in color shows the influence of temperature and duration of burial, that is important in oil exploration. And lamni may be related to astronomical days, nights, and tides, as we do with the corals. So they can give us chronological account of the past geological time period. And in this way, we can say that study of small organism is not small. And I hope that you have enjoyed the lecture. Thank you very much.